All right, everybody, welcome back to your favorite subject in the whole world, biochemistry. My name is Alfonso Flores, and I am one of your biochemistry tutors here at the University of Texas at Dallas. So before we get started, I just want to let you know that this session is being recorded, and if you wish to remain anonymous, please do not state your name, discuss your grades, or say anything that may potentially identify you. All right. Are there any questions about that disclaimer? Okay. All right, so for Dr. Taylor's section and Dr. Marsh's section, can't believe I didn't call her name out the right way. It was right there in front of my face. All right, sorry about that. Okay, so for both of you, we're gonna cover this first part, protein's primary structure, and the sequencing and the protein secondary structure. On Tuesday, March 3rd, you'll cover the rest of the secondary, tertiary, and quaternary structures. So we're only going to do part 1 of the Thursday class. We're not going to cover the whole thing. And the same thing for Dr. Marsh. Okay, so the topics we'll be discussing today are determining the protein's primary structure and sequencing, as well as the protein secondary, tertiary, and quaternary structure. Okay, so the point of the last 2 lectures that you guys just had was to answer the following 2 questions. How can we find out which amino acids are in this protein, and how can we find out how they are arranged? So the whole point of your last few lectures was to say, hey, let's say that we have this polypeptide or this protein. How can we identify, or how can we figure out what type of amino acids are in here? Does that make sense? So you have a protein, now you want to know what type of amino acids are in this protein. How can you figure it out? Yes?
Based on how you arrange like polar, non-polar, maybe?
Okay, so we can figure out if there's any polar or non-polar residues. What's a scientific way that we can do it though? I guess a more precise method of doing it. If you have a protein, how can you figure out what type of amino acids that you have on it? Sequencing. So that's what we're going to discuss. If you want to figure out the type of amino acids that you have on this protein right here, we're going to call this protein A. If you want to figure out every single one of these amino acids, you got to sequence it. Does that make sense? Any questions about that? Okay. So for those of you in Dr. Marsh's class, you'll recognize this outline. So we are going to determine we're going to do analysis of amino acid composition, and that's pretty much getting a protein, figuring out what type of amino acids are in that protein. And then we'll also do the rest of this, determination of amino acid sequence by Edman degradation and mass spectrometry. So this part right here tells you what kind of amino acids you have, what kind of amino acids you have in a protein, and this second one right here tells you how they are arranged. So from this lecture right here, for both of you that are in Dr. Taylor and Dr. Marsh's class, these are the 2 main questions that you should be able to answer as soon as you are done going through these PowerPoint slides. All right, so how is the amino acid composition of a protein determined? The first thing you got to do is you got to get your protein. Where can you find a protein? Pretty much anywhere, right? If you have a plant, like a little flower, there's a bunch of proteins inside of that flower that help every single process occur. Where else can you find proteins? Food. Where else? One more. What was that? Yeah, in your own cells. So you can find proteins pretty much anywhere. And the first step to determine the amino acid composition of a protein is to denature it. In this example, you are using 6 normal hydrochloric acid. Normal is roughly equivalent to molarity. So if you ever see N, you can just pretty much take that as molarity. If you put a protein in a very acidic solution, what is going to happen to it? if you put a protein in a very acidic solution, what is going to happen to it? It's going to be denatured. What does that mean?
It'll lose all the structure except for the primary.
You're going to break down its structure, and you will lose the whole structure except for the primary. So this is your protein. This is protein A again. And if we were to put this in 6 molar hydrochloric acid, this is what's going to happen to it. It's going to become denatured. Does that make sense? Are there any questions about that? Is anybody lost so far? Okay, so if you put this in a very acidic solution, you will denature it, and you'll be left, you'll go from the tertiary or secondary structure to the primary structure. What is the primary structure of a protein? It's the polypeptide chain, which is the sequence of amino acids. Good job. What is it when you degrade? I just said it. What is it when you destroy the primary structure? That's called degradation. You might want to remember that. So there's denaturing and there's degradation. Denaturing gives you what structure of a protein? Primary. How do we break the primary structure? Degradation. So these are all the individual amino acids right here. And that is achieved through degradation. All right, so if you want to find out what type of amino acids are in your protein, you have to denature it. Then, after you denature it, you got to take every single one of those amino acids and you got to break them into their individual, or you got to take the amino acids that are in the polypeptide and break it using Edman's reagent or orthophthaldehyde, however you say that. I forgot how to say it. But yeah, you got to take either of these 2 reagents right here, and you have to break this polypeptide chain using these 2 reagents. I'll get one for you after class. Go ahead and sit next to someone who has it. Okay, so the first thing you got to do is you got to denature the protein. Then what do you got to do to that polypeptide? Degrade it using what reagents? Edman's reagent or orthophthaldehyde, something like that. And then after you degrade it, what are you left with? The individual amino acids. And what do you do to those individual amino acids? You use HPLC to separate. After you've used Edman reagent, you have derivatized amino acids. All right. So, number 1, to determine the composition of a protein, we must first denature it, and these are some ways in which this can be accomplished. So for your exam, you'll want to know that you can denature proteins by, that's not what I wanted to use, yeah. You can denature proteins by exposing them to high temperature, exposing them to a crazy pH, like a really high pH or a really low pH, changing the salt concentration, and putting them in solvents. You'll definitely want to know that. And this is the slide from Dr. Marsh's class, which pretty much tells you the same thing. Heat, acids or alkali, surfactants, that would be your solvents. And then there should be salt concentration here. It is not, but it's one of those. So these are the ways in which you can denature a protein. Are there any questions about that? Yes?
Not necessarily denaturing, but degradation. Is Edman's reagent the main way for degradation, or are there other ways we need to keep it?
There's other ways that you can degrade a protein, but for your exam, you'll most likely see Edman's reagent, and you'll also see other enzymes which can break down proteins.
Okay.
So it's not just chemically, but you can also use enzymes to degrade a polypeptide. Yes?
Just a quick, like on that left slide, did you get that from Taylor's class?
I got it from Google. Yeah. All right, you got a question?
Yeah, so Edman's, never mind.
Okay. Are there any other questions? Okay. So after you've denatured the protein, this is what you're left with, the primary structure. And just so you guys know, you can always renature the protein. Renaturing does what to the denatured protein? It doesn't put the amino acids back together because you had that polypeptide. So the amino acids were always together to begin with. But what structure do you gain back?
Secondary, tertiary.
The secondary and tertiary structures. Good job. Oh, and you might want to pay special attention to this, the reversibility column. If you see with heat, the minus sign means that you're not very likely to be able to renature it if you denature it using heat. Does that make sense? So if you use heat, you have a very low likelihood of renaturing that protein.
Where would you see the minus sign?
What was that?
Where would you see the minus sign?
Oh, it's right here.
Oh.
Yeah.
So I mean, like, if you were to give us something, would
Yeah.
you just like tell us, like, a question?
Well, he might even, so an example question would be, which of these following methods of denaturation is the one that is least likely able to restore the protein? Or if you were to use temperature, are you more likely to be able to renature that protein? And the answer is no.
So temperature and pH are like some of the main ones.
Yeah. And pretty much all the other ones, you can always renature them. Oh, zoom in?
Yeah.
Sure. Is that enough? Okay.
Excuse me?
Yeah.
You say that, so temperature is at least likely, and then after that is pH.
Yeah, so if you denature a protein using temperature, then you are not very likely to be able to reverse that denaturation and get back that secondary or tertiary structure. So in other words, temperature is a very powerful way to denature a protein. An example of that your professor may have given you in class is whenever you're cooking eggs and you go from that clear white to you go from the clear protein to like the solid white, that's an example of you denaturing it, and then it doesn't like spontaneously turn back to the clear form, right? Eggs don't uncook themselves. Yeah. Okay, so true or false? Denaturing a protein results in the loss of the quaternary, tertiary, and secondary structure.
Oh.
That is true. It's okay. So yes, if you denature a protein, you will lose the quaternary, tertiary, and secondary structure. What about this one? Denaturing a protein results in the loss of the primary structure. Is that true or false? False. How can you lose the primary structure? Degradation. What are some ways in which you can degrade a protein? Edman's reagent, ortho something aldehyde, and enzymatic cleavage.
Did you say those Trypsin, denature? Is that what you said?
No.
You said degrade.
Degradation. Okay, so number 2, after denaturing the protein, we must pull off its amino acids one by one. How can we do that? And that is Edman's reagent. So Edman's reagent takes off every single amino acid one by one. All right, so for the first question that is on your sheet, which of the following factors will influence the native conformation of a protein? What is the native conformation of a protein?
The natural.
The natural, I like that. You can also take native conformation to be a fancy term for tertiary structure. So which of the following factors will influence the native conformation or the tertiary structure of a protein? Will the pH affect that tertiary structure? Yes. What about the concentration of salt in solution? Yes. What about the sequence of the protein?
Yes.
Of course. So every single polypeptide, every single protein has a different composition of amino acids, and that composition of amino acids dictates its structure. Are there any questions about that? Every single polypeptide has a specific sequence of amino acids, and that specific sequence is what gives the protein its shape, its tertiary structure. Does that make sense? Okay, so is C true or false? C is true. Good job. What about D, all the above? Yeah, that's the correct answer. Are there any questions about that? Any questions at all? Okay. So we've already discussed how we can denature a protein, and now we can break down this protein to get into the amino acids using this reagent, ortho something, and Edman's reagent, which is phenolysothiocyanate. On the exam, they may give you this name, or they may give you this name. They may not say Edman's reagent. So make sure that you know what Edman's reagent is. And that is this guy right here. Does that make sense? Any questions? Yes.
What's the difference between the first one and second one? How do they cleave?
So the question is, what's the difference between ortho something dialdehyde and phenolysothiocyanate, or Edman's reagent? Well, Edman's reagent reacts by interacting with the N-terminus of polypeptide, whereas with ortho something dialdehyde, I'm not too sure. I didn't see it on your PowerPoint slides. But this is how Edman's reagent works. So if you see this right here is a polypeptide chain. So this is that denatured protein that we had. And look what Edman's reagent does. This is the reagent right here, and it's going to react with this N-terminus right here. So once it interacts, it cyclizes. You don't have to really know this mechanism, but what you do have to know is that if it takes this whole amino acid right here, it takes this whole part with it, and that's that right there. So effectively what Edman's reagent did is it took this polypeptide chain, and it can go all the way till here, and it takes this amino acid, then it takes this one, then this one, then this one. Does that make sense?
So it just goes down that.
Yes, it goes down and it only attaches to what?
N-terminus.
The N-terminus. And this Edman's reagent works with every single amino acid. Does that make sense? So it doesn't matter what composition of amino acids you have, it works with every single one of them, and I think it's limited up to 30 amino acids. It should say on your PowerPoint slides, but it can't fully degrade a polypeptide chain. It can only do chunks of 30, yes.
So that all on the right, that PTH derivative, that's one amino acid with the Edman's reagent.
Yes. So what he said was that this PTH derivative is the amino acid that was just taken off from the polypeptide chain and attached to Edman's reagent. That is correct. Are there any questions about that? Okay, and so this right here, this is your derivatized amino acid. This is your derivatized amino acid, which is what you have right here. All right, and so after you've done this and you break off a bunch of those amino acids, you can actually take that composition and figure out how many amino acids or what's the relative quantity of those amino acids in your polypeptide chain. And you do that through high performance liquid chromatography. And I don't think that you guys discussed that in your lecture. So yes, so we won't be going over that in this review. But yeah, so the area under each curve represents the relative quantity of each amino acid, which you'd have to like compute using a program. And I'm pretty sure you're not going to be asked to do that on your exam. That'd be very difficult. But does this tell us the sequence of amino acids? If you were to break up the polypeptide, does that tell you the sequence? No, it does not. So now that we've collected all the amino acids, how can we find out how they are arranged? The answer is, let's start with the same protein, but we're going to treat it differently to find out the exact sequence. So we took that protein, we broke it up, and we found the composition of amino acids. Are we okay with that? Now we're gonna take that same protein, like the one that's not broken up, but the same kind, and we are going to, we're gonna degrade it and we're gonna sequence it. And so that's this part right here. So you take the protein, you break and block disulfide bonds, cleave it with peptides and proteases, and sequence specific reagents, isolate the peptides, sequence by Edman degradation, and assemble the full sequence. So this part is pretty involved. Alright, so the first thing we gotta do is we gotta break up the disulfide bridges of the polypeptide. What amino acids make up disulfide bridges? Cysteine. So here you have the polypeptide, and there could be, if we were to zoom in right here, if we were to zoom in right there, we could see one polypeptide chain and another polypeptide chain, and a cysteine residue coming out of it, and then another cysteine residue coming out of it, and they're bound together. They're covalently bound together. What type of bridge is this called? That's called a disulfide bridge. Okay, so the first thing we gotta do is we gotta break up the disulfide bridges on this protein, and we do that using DTT, dithiothiol. You might wanna know that. Very important. It's also called Cleland's reagent. So know DTT, Cleland's reagent, and dithiothiol. So the disulfide bonds are broken by reduction. What does DTT do to disulfide bonds? It breaks them by what? By reduction. And that's exactly what goes on here. Well, this shows you mild oxidation, but if we were to the reverse, so we were to go from here up, what's happening to the disulfide bridge? If we go in the upwards direction, it's being reduced. So this would be reduction. And downwards would be oxidation. Okay, oh, and there's one more that you'll want to know. Beta mercaptoethanol, which is that one right there. So you want to know that Cleland's reagent and beta mercaptoethanol can be used to break disulfide bridges, break disulfide bonds, which is essentially happening through a reduction reaction.
So both of them do the same thing?
Yeah. Maybe one of them is more mild and one of them is like stronger. I don't exactly know the difference between both. Are there any questions about that? Any questions at all? Yes.
So you're saying that beta mercapto, that one also breaks disulfide bonds through reduction?
Exactly. Perfect. Just like that. So DTT and beta mercaptoethanol both reduce disulfide bonds, and so they break them. Okay, so number 2 on your sheet, the Edman reagent, phenyl lysothiocyanate, reacts with which amino acid?
As in the
C?
So Edman reagent reacts with which amino acid? All of them.
It's an N-terminal, so yeah.
Yes, so it reacts with the N-terminus of every single amino acid. So the answer is not C, because C is corresponding to N, which is what amino acid is this? Asparagine, good job. Asparagine, I'm pretty sure that's how it's spelled. What about C, what amino acid is that? Good job. What about W? Tryptophan, close.
It was Edman reagents, and then what was the other one? Or
So the other one is ortho something dialdehyde. Ortho this dialdehyde. What does the Edman reagent do to the polypeptide chain? Denature it or degrade? Degrade. What denatures the protein?
Anything involving heat, like acidic or temperature or?
pH,
Heat,
solvent.
pH, salt concentration, and I don't remember it. Like,
There's four.
Oh, the solvent?
yeah, the solvent. And that would be, okay, that would be something like tween, triton. So these right here would be pretty good solvents. I don't know what they're gonna tell you on the exam, so like anything's fair game. Okay, so number 3, the Cleveland reagent dithiothiotol is used to do what?
Reduce disulfide bonds.
Reduce disulfide bonds to sulfhydryls. Good job. Are there any questions about number 2 or 3? Okay, so the follow-up to the first question, what would happen if the N-terminus of a polypeptide was blocked through acetylation? So let's say we have a polypeptide and the N-terminus was blocked through acetylation. What is acetylation?
It's where you neutralize positive.
Okay, so that's true, through acetylation of the N-terminus, that's exactly correct. You do neutralize the charge. Okay. What type of modification is acetylation?
The addition of
Addition of acetyls.
It's the addition of an acetyl group. Good job. But I'm looking for like the textbook answer. It's a post-translational modification. You guys might want to know that. Acetylation is a post-translational modification. All right. So if we were to acetylate the N-terminus, would we still be able to use the Edman reagent? Yes or no? The Edman reagent reacts with what? The N-terminus. And if we block the N-terminus, can we still use the Edman reagent? No. Good job. Are there any questions about that? Yes.
So if it blocks the first N-terminus, we can still use it for the other amino acids?
No, because the Edman reagent only works, it works from like the outside inward. So like if this right here is our polypeptide chain, let's say this right here is the N-terminus. So this right here is the N-terminus. If we block it by putting an acetyl group on it, then the Edman reagent will not react with it.
So it only works with NH3.
Yes.
NH2, not the peptide form itself.
Exactly. So there are let me draw this right here and I'll show you. So this right here is the structure of a polypeptide chain. So this right here would be the N-terminus. Okay, and this goes on and on and on, right? Because it's a polypeptide chain, so it can go on forever. Okay, so it goes on forever. The Edman reagent only reacts with this, the NH3+. You see this right here, this NH? It's not going to react with this. So that's what you were asking, like, if we block this one, then would the Edman reagent react with the nitrogen here? Or the nitrogen here? It won't. Does that answer your question?
Yeah.
Okay. What are some ways that we could post-translationally modify the N-terminus? Methylation. What else?
Phosphorylations.
Never heard of phosphorylation.
methylation formal.
What was that?
You add a formal.
I don't
Marsh
remember.
called it
Cause like on Marsh's slides, there's like 5 methods of post-trans.
Yeah, okay.
yeah.
But not every single amino acid can be post-translationally modified. So Like for example, you could modify the N-terminus by putting an acetyl group or a methyl group on it. But what about like the side chain of valine? Would you be able to post-translationally modify that by putting an acetyl group on it? Not really. Okay, so no, if we blocked the N-terminus through acetylation, we would not be able to use the Edman reagent on it. Okay, so now that we have a polypeptide chain without any folding due to disulfide bonds, we can digest it with different enzymes so that we can break it up into smaller parts which are easier to sequence. So you're going to want to know all of these enzymes for your exam. So you want to know trypsin, V8 protease, chymotrypsin, pepsin. I don't know about thermolysin. And you're going to want to know the methods to chemically cleave polypeptides. So let me reiterate. So far, all we've talked about is getting a protein and breaking up the disulfide bonds. Are we okay with that? Now we're going to take this protein. So we're going to take this protein and look how huge it is. So in order for us to be able to sequence it efficiently, we have to break up this protein here, 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 and here. Does that make sense? You got to break up the protein. How can you do that? You can use enzymes. That's the best way to do it. So,
why are we breaking it again?
I'm sorry, what was that?
Why are we breaking it?
So the reason why we are breaking up this polypeptide is because we want to find out what? We want to find out the exact sequence of a protein. So just imagine this. Imagine you find or you discover a new protein and you want to know like how it's arranged in terms of its amino acids. You would break it up into smaller chunks and then analyze those smaller chunks. Does that make sense? Okay. So trypsin, here's what you're gonna want to know. It cleaves after arginine and lysine or aminoethylcysteine. V8 protease cleaves after glutamate and also after aspartic acid, and that's it. So this one, V8 protease, cleaves what type of amino acids? Acidic. Good job. So V8 protease cleaves after acidic amino acids. And we'll go over what that looks like. Cleaves after acidic amino acids. What about chymotrypsin? After what type of amino acids does it cleave?
Tryptophan, tyrosine, glycine.
So we have phenylalanine, tryptophan, tyrosine, and leucine. What's the similarity between phenylalanine, tryptophan, and tyrosine? They're aromatic. So you can say that chymotrypsin cleaves after the aromatics. Plus what? Plus leucine. All right, and pepsin, phenylalanine, tyrosine, tryptophan, so also the aromatics. But there is something that you're going to want to know about this one, and it is, let's see, there's an exception somewhere. Okay. So I don't see any exceptions on this PowerPoint slide, but if there are, I'll send out an email to you guys and I'll let you know.
Talking about like the proline,
Where is it at?
at the very bottom, it says that all five enzymes.
Ah, thank you. Perfect. Appreciate it. Okay, look, this right here, thank you for pointing it out. I really appreciate that. This right here is going to save your lives. Usually non-cleave bonds when protein follows the amino acid. So let's say that we have an amino acid and it is P-gabal. Give me an acidic amino acid. No, ED. That's good enough. let's say this right here is our polypeptide. If we were to use chymotrypsin, how many cleavages would we have on this polypeptide sequence? None, right? Because there is a leucine here, but what follows it? Proline. So because there's a proline, the chymotrypsin will not cleave after this leucine. Does that make sense? Are there any questions about that?
Can you say?
Sure. So this right here is our polypeptide sequence. And let's say that we were to digest it with chymotrypsin. So we're gonna digest it with chymotrypsin. What or after what amino acids does chymotrypsin cleave? After phenylalanine, tryptophan, tyrosine, and leucine. So if you see here, we have a leucine. So if you know that chymotrypsin cleaves after leucine, you'll immediately want to cleave it. But there's an exception. If proline follows that amino acid, you cannot cleave. Does that make sense? Are there any questions about that? Yes.
If we can't cleave it anywhere, is that 0 fragments or 1 fragment?
That would be 0 fragments. Yes. So the question was, if there's a proline here, there will be will there be 0 fragments? And the answer is yes, there will be. Yes.
And that's for all of them, right? The exception, or is it just for?
That's what it's implying here. That usually non-cleave bonds when proline follows the amino acid. So I would say yes, because it's in the PowerPoint slide. You can't argue with the PowerPoint slide. Okay, so these are enzymatic cleavages, and these are the chemical cleavages. And as you guys know, we've already discussed, or no, we haven't discussed any of these. But look, cyanogen bromide, that's a really good one to know. All right. So for this question right here, I believe it is number 4. Digestion of the following fragment with trypsin will produce how many amino or how many fragments?
2.
Where will it cleave?
Let's see.
So we're digesting it with trypsin, and trypsin cleaves after R and K. And aminoethylcysteine, there's no aminoethylcysteine. So
So it has to say that? How would you know that? If you see the C in a sequence like that?
if you see a C and they tell you that on the question that there's an amino ethyl group on it, then you'll know that cysteine has been modified that way. Okay, so it cleaves after R and K. I don't see any R's, but I do see a K here, and I see a K here. So how many fragments will we have? Why 2? I thought that it cleaves after this K as well. Proline follows it, so there's no cleavage here. Good job. So it'll only cleave where? Will it cleave to the left of K or to the right of K? To the right, because it only cleaves after lysine. And it's the same for all of them. Are there any questions about number 4? Okay, so for number 5, digestion of a polypeptide with V8 protease. For those of you who came in and didn't grab a sheet, don't worry, I will get you one as soon as we're done. Okay, so number 5, digestion of a polypeptide with V8 protease and trypsin will lead to the following fragments. What is the correct sequence of the polypeptide? What is the correct sequence? So let's do this. Let's first find a small fragment. After we found the smallest fragment, we're going to try to find that small fragment in the larger ones. Does that make sense? So here we have LVGK. And if you see here, we also have LVGK over there. Does that make sense? So whenever you're given a problem like this, look for the smallest fragment and then search for that smallest fragment in a larger one. So here we have LVGK, and then we have LVGK as well over here. What follows after this K? AE. And where else do we have AE? We have it up top right here. And what follows after E? FS. So then what is the correct sequence of the polypeptide? We can start off with LVGK, and then what? AE, and then FSGITPK. Are there any questions? Yes. Sure. So I'm going to do number 5 again. So let's erase everything, start from the beginning. So if you're given a problem like this on the exam where they tell you that you took a polypeptide and you digested it using these 2 enzymes. So they told you that you digested the polypeptide with these 2 enzymes, and they ask you for the correct sequence of the polypeptide. The first thing you want to do is what?
Look for the smallest fragment.
Look for the smallest fragment possible in either of these 4 that you're given. What's the smallest fragment that you have there? LVGK. So after you found your smallest fragment, what do?
Write it down.
You write it down. That's one thing you could do. But after you found the smallest fragment, what should you do?
Find a bigger fragment.
Find a bigger fragment that has that small fragment in it. So here's a big fragment, and if you see here, you have LVGK. It's okay that it's written backwards, because polypeptides, they can, you can rotate them. Does that make sense? Are there any questions so far? So if you're given a few digested sequences, find the smallest sequence, and then that smallest sequence, find it in a larger sequence. And that's how you're able to piece together the correct sequence of the amino acids. So we know that we have LVGK over here, and we have LVGK over there. What follows after this K?
AE.
A and E, which is also here. And then you know that after E you have FSGITPK, which is over there. So then the correct sequence would be what? LVGK. What comes after the K?
AE.
AE. What comes after the E? FSG. ITPK. Good job. You're good at puzzles. I can see that. Okay. Are there any questions about that? Yes.
So it doesn't matter like which order you use EPA versus Trypsin?
Well, here in the question, they were already digested, but as long as you get like the correct sequence, like just in the correct order, then you should get the right answer. Okay, so that's number 5. And number 6, CNBr, cyanogen bromide. Cyanogen bromide is commonly used to generate peptides from proteins for sequence analysis. After which amino acid will it cleave? Methionine. Good job. We didn't even go over that. Jeez, you guys are on top of it. I messed up. I should have told you. But it cleaves after methionine. So cyanogen bromide only cleaves after methionine, and you're going to want to know it for all of these as well. So what's the answer for number 6? D, none of the above. All right, are there any questions? Yes.
So, on the workshop, you learned that, are we supposed to replace it, when using that one, are we supposed to replace the methionine with, I think it was homoserine lactone? She put that in for the workshop, so I don't know if that's something we need to know for the exam.
Well, I've never heard of that,
Okay.
and in all the time that I've tutored biochem, I've never heard of methionine being replaced with, what was it again?
Homoserine lactone.
Homoserine lactone. I think that was specific to your problem set.
Okay.
Yeah, I don't think it was specific to these slides. Who's your professor, if you don't mind? Okay. If it's not in her slides, I wouldn't worry about it. Yeah. Are there any other questions? All right. So we've already spoken about how to block disulfide bonds. What reagent can you use to break disulfide bonds? DTT. What other reagent could you use? Yeah, beta mercaptoethanol, good job. Okay, and after you've broken those disulfide bonds, how can you cleave that polypeptide chain into smaller polypeptides?
Trypsin, EA, Chymotrypsin.
So what type of cleavage is that when you use enzymes?
With time.
Yeah, it's enzymatic cleavage. Yeah. Okay, so you can also chemically cleave it, and you would be using these reagents right here. Are there any questions about all this stuff that we've covered so far? No questions? Okay, so moving on. So we're not gonna discuss Edman degradation because we've already done that. What does Edman degradation do again? It degrades polypeptides by doing what? It binds to what? The N-terminus of which amino acid? All of them. Good job. Okay, so now number 2. So this is going to be from your Thursday lecture or your Wednesday lecture if you're in Dr. Taylor's class.
You're on top of it.
Yeah? Thank you. I'm on top of it. Okay, that's a microphone. All right, so for protein structure, sorry, was there a question anymore? Okay, yes. Oh. All right, so the primary structure of a protein is what? What's the primary structure of a protein?
The amino acids.
Just the amino acids, and how are they bound? They're bound covalently. And does the primary structure include disulfide bonds? Yes, it does. The primary structure includes disulfide bonds. You'll definitely want to know that. Does anybody know what this translational part is? So do you guys know what DNA is? Okay, that's a good start. So you have DNA, right? How do you can make mRNA out of DNA through transcription? You remember learning that? Good job. So you transcribe DNA and you end up with what? mRNA, perfect. Okay, so this is mRNA. And then what do you do to mRNA to turn it into a polypeptide? Translate. Okay, so then when you translate mRNA, you get a polypeptide. After you have this polypeptide, or after you've made this polypeptide, what can happen to this polypeptide? It can be post-translationally modified. So post-translational modifications can occur. So an example of that would be, imagine you have a cysteine residue here and a cysteine residue right there. Some other enzyme is going to take this polypeptide and then connect these two cysteine residues together. Does that make sense?
Yes.
This is just one type of post-translational modification. Does that make sense? What if we had a hydroxyl here So if we had that hydroxyl there on this polypeptide chain, it would be here. How can we further modify this hydroxyl? What amino acid residue is this? Serine, good job. So how could you post-translationally modify serine? How can you post-translationally modify serine? You can, I've never heard of hydroxylation for serine, but you might be right. What about, I'm sorry, what was that? So you can't oxidize this hydroxyl any further, but what about phosphorylating it? So we grab another enzyme. This enzyme uses ATP, releases ADP, and now look at this polypeptide chain.
Why are we doing this?
Yeah, this is important. It's definitely gonna be on your exam.
So like, why is it being phosphorylated?
Oh.
That's
That's what I was trying to
I wasn't saying that.
You were saying, is this important? Yeah, this is,
All
no.
of this.
You're definitely gonna wanna know this. Okay, so phosphorylating it is modifying the polypeptide, and that affects the function of this polypeptide. It can make it work better, or it can make it work worse, or it can just change the shape of the polypeptide. So these post-translational modifications are important for all the polypeptides that are pretty much in your body. Does that answer your question? It answers your question, and it kind of answered yours. Okay, so yes, this is important for both of you. Okay, so the primary structure is the amino acids linked together, and does it include disulfide bonds? Yes, it does. What type of modification is it when you form disulfide bridges?
Post-translational.
Yes. Whenever you form disulfide bridges, that is a post-translational modification. What's another type of post-translational modification?
Phosphorylation.
Perfect. Phosphorylation. What type of enzyme does phosphorylations? A kinase. But what is the enzyme classification of those enzymes? What is the classification of an enzyme that transfers one phosphate to another molecule? Transferase. What is the enzyme classification for transferases? The number. Good job. Number 2. Okay. So the secondary structure of a protein is the alpha helices, the beta sheets, and the beta turns. So you have alpha helices, beta sheets, beta turns. And whenever you have something like an alpha or a beta, alpha, beta, that is something called a supersecondary structure. So basically, supersecondary structures are made out of what? Alpha helices and beta sheets, but another way to think about it is that supersecondary structures are made out of just secondary structures. Does that make sense? Are there any questions about that? Okay. So these are the 4 levels of the protein organization. This right here is an alpha helix, and this is a beta sheet. Essentially what you have is a polypeptide, and every single polypeptide has a specific sequence of amino acids, and it's that specific sequence of amino acids that makes, like this region right here, turn into a helix. I'm not good at drawing, so please forgive me. Thank you. Okay, so this right here is an alpha helix. So what's happening is every single one of these amino acids is turning into a helix, or they're forming a helix. That's a crappy drawing. But yeah, so essentially they're forming into this alpha helix right here. Does that make sense? And it's because of the specific sequence of amino acids that they can do that. And then you can have something called a beta turn. So this right here would be your alpha helix. And then this part right here could be a beta sheet. And this part right here could be these amino acids over there. Does that make sense? So all of the secondary structures are due to what?
No, what are they?
How do these alpha helices and beta sheets convert? Or how do they get turned into alpha helices and beta sheets? So the answer is that it has everything to do with the specific sequence of amino acids. So it's the specific sequence of amino acids that allow the formation of the alpha helix and the beta turns and the beta sheets. Okay, so this polypeptide, which is the primary structure, so this right here is the primary structure, and within the primary structure, you'll have regions that get turned into alpha helices and beta sheets. So this will be another turn, and this will be another beta sheet. Okay. So why do these polypeptides get turned into alpha helices, beta turns, and beta sheets? Because of the specific amino acid sequence. That's a really important thing for you guys to know. So you're gonna want, bless you, you're gonna want amino acids here that can bend easily. What type of amino acids do you know that can bend easily? What about glycine? So this is glycine right here. And the R group is just a hydrogen. So whenever you have glycine in a polypeptide chain, because it's so small, it's able to bend easily. And that's the type of amino acid that you'll find in beta turns. And there's specific amino acids that you're more likely to find in alpha helices, and there's specific amino acids that you're more likely to find in beta sheets. Does that make sense? Are there any questions about that? There's specific amino acids that end up in alpha helices, and beta turns, and beta sheets. Okay. So after this polypeptide folds and gets turned into, this part turns into alpha helices, and this part turns into beta sheets, and then this one into another beta sheet, and it turns and folds even further, you get the tertiary structure, which is pretty much this, a big globular protein. This is when you've achieved your three-dimensional shape. Okay, so why do these secondary structures even form? What is it due to?
Specific,
Yeah.
specific amino acid sequences.
All right. And so you are more likely to find one kind of amino acid in an alpha helix, and you're more likely to find one type of amino acid in a beta sheet. Does that make sense? All right. So that's because the polypeptide chain is only able to twist and turn in certain directions. So with every single amino acid, you allow a certain form of twisting, and this is really important for you guys to know. So the primary structure of a protein is the what? Polypeptide. This primary structure contains a CN angle and two bonds which are limited in rotation despite their single bond characteristic. Draw it below. Okay, so this is the primary structure contains a omega angle. All right, and that's fixed. Does that not look like omega?
I don't know what omega looks like.
No. Omega is a Greek letter, I'm sure. Am I right?
Yeah.
Okay. That's omega. All right, so the primary structure of the protein is the polypeptide. And the primary structure contains an omega CN angle, which is fixed. That's really important for you to know. The omega angle is fixed. And two bonds which are limited in rotation despite their single bond characteristic. Draw it below. All right, so I'm going to draw polypeptide here. And I'll start with the N terminus. And what we're going to do is we're going to identify every single angle. So you have NH3 plus.
You're just drawing any random one?
Yes. All right, so this is just a random polypeptide chain.
Oh, you're just drawing one.
Mm-hmm. Yeah. All right, and it goes on and on and on because it's a polypeptide, right? It's just a bunch of amino acids put together. So here's what you got to know. This part right here, this has a partial double bond characteristic. So this right here has a partial double bond characteristic. So because it has a partial double bond characteristic, is it able to rotate? So can we rotate this part right here? Let me zoom in. Can we rotate this part right here like that? Yes or no?
No.
Damn it. I had a really good structure and I messed it up. Okay. Yeah. So because this nitrogen is resonating and so is this oxygen, the carbon, it's not able to rotate, and this right here is your omega angle, and what you gotta know about it is that it is fixed. Because of the resonance, the partial double bond characteristic. So we say that the N and carbonyl carbon, that is the omega angle, and it is fixed.
So it cannot rotate?
It cannot rotate, not even if it's in its wildest dreams.
Which,
Cannot rotate.
what's the omega here?
The omega, it refers to the angle.
Okay, so just like where it's not able to rotate?
Right.
Okay.
Okay, and so then you also have the nitrogen alpha carbon angle, and I believe that is the phi angle. Let me check. Yes, that is the phi angle. Yeah. So this right here is the phi angle. It has a single bond characteristic, so therefore it can rotate. So this is your phi angle, or phi. Do some of you say phi instead?
Feet.
Who says phi? You're cool.
I'm like, who's a tee?
Okay, and so this angle right here, it can rotate, okay? You see how it's a single bond? It can rotate. What about this bond right here?
Wait, what is phi look like?
It's this right here. This is the lowercase phi.
Oh.
Yeah, and this right here is omega. And this angle right here is called what? This is the psi angle, which looks like that.
It's like a trident.
Yeah, it looks like a trident. Or maybe that's how I draw it.
No, I think you're right.
Okay, so can the phi and the psi angle rotate?
Yes.
Yes, they can. What is the name of the angle between the nitrogen and the alpha carbon? The nitrogen and the alpha carbon? Phi. Good job. It's phi. And what about the angle between the alpha carbon and the carbonyl? Psi. That's bad. Okay, can these rotate right here? The nitrogen alpha carbon and alpha carbon carbonyl, can these rotate?
Yeah.
Yes, big time. They can rotate, but they can't rotate fully. So like you can't rotate them any way that you want. Because look, If you were to rotate this angle right here, so I need you to pay special attention to this angle right here, or this angle. If I were to rotate this angle, so I rotate that single bond, this R group right here could clash with any other R group that's in the chain. So if we rotate about the single bond any way that we want, molecules can start interacting, which is I guess that's like another way of saying that they start getting in their own way. So there is an image that I want to show you, like right over here. Okay. So this nitrogen right here and the alpha carbon, this angle right here, it can rotate, right? Yes or no? Yes, it can rotate. But if it rotates all the way, look at this carbonyl and look at this carbonyl right here. You see how they're clashing? So they're getting in their own way. And also, if you rotate about this angle over here, they get in their own way again. Okay, so for number 8, the psi angle refers to the amount of rotation about which bond in the peptide backbone? C alpha, C what? C carbonyl. So the answer for number 8 is B. The psi angle only refers to the angle between the alpha carbon, which would be this one right here. The alpha carbon has what sticking out of it? The R group. Good job. So the alpha carbon has all the R groups. So each one of these is an alpha carbon. So there is rotation about the alpha carbon and the carbonyl, and that is the psi angle. So that is the answer for number 8B. Are there any questions about number 8? Okay, so can the phi and the psi angle rotate, yes or no?
Yes.
But can they rotate in any, can they rotate any way that they want?
No.
No, they can't. I like how you said it so strongly. Okay, so the reason why they can't is because they start getting in their own way. They start their atomic radii start interacting. It's quantum physics. I don't really know that. But here's something that's really important that you guys should know and should pay very special attention to, that the phi angle 0 degrees and the psi angle 180 is not allowed. The 180 and 0 degrees for phi and psi are not allowed. And 0 degrees and 0 degrees are not allowed. Why?
Because
They get in their own way. There's like a more scientific term for it. I think it's called steric crowding.
oh yeah.
Okay. So this guy said, hey, since the omega angle is fixed, and since the phi and the psi angles vary, then this must explain why certain combinations of phi and psi angles form alpha helices and beta sheets. He didn't really say that, but he said something along those words. And he came up with this Ramachandran plot. Have you guys discussed Ramachandran plots in your problem sections?
In problem section? No.
Okay, but what about in class?
Yeah.
Okay, so you guys know how we just discussed that the phi angle and the psi angle can rotate?
Yes.
Okay. What the Ramachandran plot pretty much says is that, hey, the psi angle, when it's about, let's say, what is that, 160? No, 120? 130. When it's about positive 130, and when the phi angle is about negative 100, the structure that we see is what? Anti-parallel beta sheets and parallel beta sheets. So maybe it's too small for you to see.
What does it say at the bottom?
Where?
Is that 100?
Yeah, so that's 100 right there. So if I zoom in, since we know that the phi angle and the psi angle can rotate, what they found is that all the ones that have a psi angle of positive 130 degrees and all the ones that have a psi angle of about negative 100 degrees, all those polypeptide chains turn out to be beta sheets. Does that make sense? So if polypeptide chain has a bunch of positive 130 and negative 100 degrees in terms of the phi and the psi angles, what type of structure do you have? Beta sheets. Okay, what if you have like negative 45 degrees in terms of psi and about 100 degrees, or no, that should be negative, we'll say negative 70. What structure you're most likely to have? You're most likely to have a right-handed alpha helix or a closed ring. Does that make sense?
Yes.
So a Ramachandran plot is not something that you have to make. It was just an analytical tool that helped them find out, hey, this is why we have beta sheets and this is why we have alpha helices, because of their angles. Does that make sense? A little more? Okay, so don't worry, you won't have to make a Ramachandran plot. And with this, let's go ahead and answer our last few questions. The Ramachandran plot below shows 3 distinct shaded areas. Please use the plot to answer the following questions. I, or Roman numeral 1. The shaded region marked A corresponds to which secondary structure?
Beta sheet.
Beta sheet. So the answer for 1 is what? Okay. Sorry for the messed up numbering.
Looks great.
Okay. So for 2, the shaded region marked C corresponds to which secondary structure? That would be a left-handed alpha helix. So you see this part right here? That's a left-handed alpha helix. For your exam, they may not make you, I don't think they're gonna make you remember all these degrees. You just gotta know that a Ramachandran plot explains the certain angles that correspond to certain structures. Does that make sense? Okay. So for C is, for number 2, what is it? It's G, right?
Yeah.
A left-handed alpha helix? Perfect. And what about 3?
Right-handed alpha helix.
A right-handed alpha helix. Are there any questions about the Ramachandran plot? No? All right, with that, thank you for coming, and I hope you all have a great exam.